For decades, a single macronutrient bore the blame for our greatest health crises. We were told that dietary fat was the enemy, the direct cause of expanding waistlines and failing hearts. Grocery aisles filled with low-fat and fat-free products, promising salvation through restriction. Yet as we stripped the fat from our plates, our collective health did not improve. In fact, rates of chronic metabolic disease continued to climb at an alarming pace. This profound contradiction forces us to ask a critical question. How did we get the science of human nutrition so incredibly wrong? Today, we are tracing the historical and scientific evolution of our understanding of dietary fat. We will uncover how a widespread demonization gave way to a massive paradigm shift, ultimately recognizing the essential nature of fats for human survival and optimal health. Let us travel back to the mid-20th century to understand the origins of this nutritional dogma. In the 1950s, coronary heart disease was rising rapidly in the Western world, becoming a terrifying public health emergency. Scientists and physicians were desperate for answers to explain this sudden epidemic of heart attacks. Enter a charismatic and influential physiologist named Ancel Keys. Keys proposed what became known as the lipid hypothesis. This hypothesis suggested a direct linear relationship between the consumption of dietary fat, specifically saturated fat, elevated blood cholesterol levels, and the subsequent development of coronary heart disease. To prove his theory, Keys spearheaded the famous 7 countries study. This was an ambitious, multinational observational project that appeared to show higher rates of heart disease in countries with higher saturated fat intake, such as the United States and Finland, compared to countries with lower intake, like Japan and Italy. The narrative derived from the 7 countries study was compelling, elegant, and dangerously simple. The prevailing logic dictated that eating fat raises your cholesterol, and high cholesterol physically clogs your arteries like sludge in a pipe. Public health organizations, medical associations, and government agencies quickly adopted this stance. Sweeping dietary guidelines were drafted, urging the public to drastically reduce their intake of total fat. Dietary fat became public enemy number 1. Subjected to an unprecedented level of widespread demonization. However, science is rarely as simple as a single villain. When we remove a major macronutrient from our diet, we must replace it with something else to maintain our daily caloric intake. The food industry responded to the new low-fat mandate with remarkable speed, replacing naturally occurring fats in foods with refined carbohydrates and heavily processed added sugars to maintain flavor and texture. This massive substitution had profound, unintended consequences for human biology. To understand why, we must look at metabolism. Metabolism is the complex set of life-sustaining chemical reactions within our cells that convert the food we eat into usable energy. When we consume highly refined carbohydrates and sugars, our bodies rapidly break them down into glucose, causing sharp, immediate spikes in blood sugar. This rapid increase triggers the pancreas to release insulin, a powerful hormone that regulates blood sugar but also actively promotes fat storage. Dietary fats, on the other hand, have almost no impact on insulin levels. By replacing satiating dietary fats with fast-digesting carbohydrates, the low-fat era inadvertently fueled a physiological cycle of constant hunger, frequent overeating, and widespread metabolic dysfunction. We were systematically eating less fat, yet society was growing progressively sicker. As the decades passed, independent researchers began to notice glaring flaws in the original lipid hypothesis. The 7 countries study, while historically significant, was purely observational. It could show a correlation between 2 variables, but it could not definitively prove causation. Furthermore, critics meticulously pointed out that Keys had selectively chosen countries that neatly fit his hypothesis, while omitting robust data from populations like the French or the Maasai, who consumed high amounts of saturated fat but experienced remarkably low rates of heart disease. A profound scientific paradigm shift was slowly taking shape in laboratories around the world. Rigorous randomized clinical trials and highly advanced biochemical analyses began to dismantle the monolithic, simplistic view of fat. We learned that dietary fat is not a single, uniform substance to be feared. It is an incredibly diverse category of molecules, each with vastly different and highly specific physiological effects. To truly grasp this shifting understanding, we must examine the molecular architecture of lipids. Fats are composed of carbon, hydrogen, and oxygen atoms linked together in long chains. The specific length of these chains and the exact type of chemical bonds between the carbon atoms determine precisely how the fat behaves inside our bodies. Saturated fat has no double bonds between its carbon atoms. It is fully saturated with hydrogen atoms. This rigid molecular structure makes it solid at room temperature, like butter, tallow, or coconut oil. For a long time, all saturated fat was viewed as equally harmful to the human heart. However, modern research indicates that the health impact of saturated fat is highly complex. It depends heavily on the specific food matrix it comes from and the individual's overall dietary pattern. For example, the saturated fat found in fermented dairy like yogurt or kefir behaves very differently in the body than the saturated fat found in heavily processed fast food. Then, we have unsaturated fat, which contains one or more double bonds in its carbon chain. This molecular kink prevents the chains from packing tightly together, making unsaturated fats liquid at room temperature like extra virgin olive oil. This vital category is further divided into monounsaturated and polyunsaturated fats. The rigorous scientific reevaluation of these specific fats fundamentally changed our modern nutritional guidelines. Polyunsaturated fats, specifically omega-3 and omega-6 fatty acids, are clinically termed essential fats. Our bodies simply cannot synthesize them from other materials, meaning we must obtain them directly through our diet to survive. Omega-3 fatty acids, found abundantly in wild-caught fatty fish, walnuts, and chia seeds, play a profoundly essential role in human biology. They are critical structural components of our cell membranes. A cell membrane is the crucial protective lipid bilayer surrounding every single cell in your body. It regulates what nutrients enter the cell, what waste products exit, and facilitates vital communication with other cells. When our diets are sufficiently rich in omega-3 fatty acids, these flexible molecules are incorporated directly into our cell membranes, making them more fluid and highly responsive. This optimal fluidity is absolutely crucial for proper cellular function and signaling. Furthermore, omega-3s are the primary biological precursors to signaling molecules called eicosanoids, which help regulate the body's entire inflammatory response. While acute inflammation is a necessary, life-saving defense mechanism against injury or infection, chronic, low-grade systemic inflammation is now recognized as a primary root cause of many modern chronic diseases. Omega-3 fatty acids actively promote powerful anti-inflammatory processes, protecting our cardiovascular system and heavily supporting cognitive function and brain health. As our understanding of dietary fats deepened, so did our understanding of cholesterol, the other heavily maligned molecule in the lipid hypothesis. The original scientists assumed that eating dietary cholesterol directly and proportionally raised blood cholesterol levels. However, decades of metabolic ward studies have proven that the human liver tightly regulates cholesterol production. For the vast majority of people, consuming cholesterol-rich, nutrient-dense foods like eggs has a negligible impact on overall blood cholesterol levels. Furthermore, the outdated concept of a single bad cholesterol has been heavily refined by modern lipidology. We now understand that lipoproteins, the microscopic particles that carry cholesterol through the watery environment of the bloodstream, come in various sizes and densities. High-density lipoprotein, or HDL, helps remove excess cholesterol from the blood, returning it to the liver. Low-density lipoprotein, or LDL, is the type historically associated with plaque buildup in arteries. But even LDL is not inherently evil. Emerging research shows that it is the small, dense LDL particles, which are often increased by high sugar and highly refined diets, that pose the greatest atherogenic risk. Larger, more buoyant LDL particles, which often increase with saturated fat intake, are relatively benign. Recognizing the essential nature of fats also involves understanding how they interact with other vital nutrients. This brings us to the concept of bioavailability, which is the exact proportion of a nutrient that is successfully absorbed from the digestive tract and utilized for normal cellular functions. Many crucial micronutrients, which are the essential vitamins and minerals required in small amounts for healthy development and disease prevention, are entirely fat-soluble. Vitamins A, D, E, and K require the physical presence of dietary fat to be properly absorbed across the intestinal wall. Eating a large bowl of leafy greens completely devoid of fat means your body is physically incapable of absorbing the full nutritional value of those vegetables. Adding a healthy source of fat, like olive oil or avocado, dramatically increases the bioavailability of these critical micronutrients. Furthermore, we must consider the profound impact of fats on the gut microbiome. The gut microbiome is the incredibly vast and complex community of trillions of symbiotic microorganisms living inside our lower digestive tract. Emerging gastroenterological research suggests that the specific types of fats we consume can significantly influence the composition, diversity, and overall health of our gut microbiome. Diets rich in diverse, healthy fats support the proliferation of beneficial bacterial strains, which in turn produce short-chain fatty acids that nourish the gut lining, regulate our immune system, and even produce neurotransmitters that impact our mood and cognitive state. Conversely, diets high in heavily processed, artificially created trans fats have been shown to negatively alter this delicate microbial balance. The scientific evolution of our understanding of dietary fat is a powerful testament to the self-correcting nature of the scientific method. We moved from a simplistic, fear-based model that demonized an entire macronutrient to a highly nuanced, evidence-based understanding of human biochemistry. We no longer view all fats as detrimental to human health. Instead, we focus intensely on the quality, the source, and the specific molecular structure of the fat. The traditional Mediterranean diet, consistently ranked by researchers as one of the healthiest dietary patterns in the world, is remarkably rich in healthy fats derived from olive oil, nuts, seeds, and wild fish. It clearly demonstrates that a diet relatively high in total fat can be incredibly protective against chronic metabolic disease, provided those fats come from whole, minimally processed, natural sources. The widespread demonization of dietary fat was a significant misstep in public health history, largely driven by incomplete epidemiological data and oversimplified public health narratives. By tracing this historical and scientific journey, we empower ourselves to look past fleeting fad diets and sensationalized media headlines. We learn to appreciate the elegant, interconnected complexity of human biology. Dietary fats are not the enemy of the human body. They are vital, irreplaceable building blocks for our brains, our hormone production, and the structural integrity of every single cell we possess. When we consciously choose high-quality, nutrient-dense sources of dietary fat, we are not indulging a dietary vice. We are actively providing our bodies with the fundamental biological materials they require to function, heal, and thrive. The paradigm shift is complete, and the biochemical evidence is overwhelmingly clear. Understanding the true, unvarnished science of nutrition allows us to reclaim our metabolic health, one informed, evidence-based choice at a time.